interesting things. That's nice to know. Thank you. And timely, too. We see all sorts of exciting things out there, from some wonderful beetles through to some predators and other kinds of herbivores, too. And, of course, the occasional monarch. We're lucky enough to have some areas up there which are protected from deer. This is an area we inherited from Luke Nave, that we can do controlled experiments, keep the deer out, and actually try and do experiments to understand the patterns we see. Occasionally, of course, we entirely take over the greenhouse. And then we do some work in the CO2 array to understand the effects of CO2. And then we take over Dave Currow's pole barn. So why do we take over the entire upper level of UMBS? The bottom line is we're trying to see what the patterns are. We're doing experiments to understand the mechanisms by which those patterns emerge. And then the ultimate goal, of course, is to inform management to do something about these catastrophic declines in the monarch. I can't share too much about what we actually do in terms of the research, but I'm going to spend a bit of time today on a couple of the ones that I think you'll find most interesting. We might expect that the environmental change will have direct effects on monarchs, things like temperature and rainfall and so on. But it turns out that many of the most interesting influences of environmental change are mediated by effects on the host plant, the milkweed host plants that monarchs use as hosts. And even then, some of the most important ones are mediated by yet other species. This is a protozoan parasite. It's called Ophrocystis electroschira. It's related to malaria, and it's related to toxoplasmosis, right? And this is a major pathogen of monarch butterflies and has important effects on their dynamics. So, what does all of this do to monarchs at UMBS? And unfortunately, of course, the answer so far is it's not great. Since we started, things had really become seriously bad. 2013 was our first 0 year of caterpillar counts using our standardized counts. 2015 was the same. So very, very poor densities these days in our standardized counts. And in fact, they map on extremely well to what's happening in Mexico. With enough long-term data, there's a statistical technique we'll be able to use to infer whether or not what's happening in Michigan is influencing what's happening in Mexico, or whether what's happening in Mexico is determining what's happening in Michigan, or more likely, the strength of that feedback process between the two. But we'll need more data. We only have 9 years of monarch data. We'll probably need about another 9 or 10 to get the answer to that. Meantime, though, one of the things that's been worrying us the most is this increase in elevated CO2. And the reason for that is some work we did with Terry Mattiella and Rachel Vinette on the effects of elevated CO2 on milkweed. Because unfortunately, what these studies show is that under elevated carbon dioxide, the concentration of toxins, cardenolides, toxic steroids in milkweed goes down. That is horrific. And the reason it's horrific is because monarchs rely on these toxic steroids for a wide variety of things. Perhaps most importantly is that those steroids protect monarchs from the protozoan parasites. So as you get increasing concentrations of cardenolide in foliage, we see a reduction in parasite burden. And as a reduction in parasite burden, infected butterflies live much longer, they lay more eggs, they mate more often, they can fly further. And so all matrices of fitness increase with foliar cardenolide concentration. This work's been done in collaboration with my colleague Jaap de Rode at Emory University. So this kind of seems like more bad news, right? More CO2 means less cardenolide means less medicine, and less medicine means more parasites. Well, to see if that pattern holds up, we do experiments, and this is Leslie Decker starting her work with CO2. So in 2014, she stole Dave Currow's CO2 chambers and pumped some with CO2, some with ambient air, and then infected monarch caterpillars with this parasite on plants grown on either on ambient or elevated CO2. So what was the result? Well, we know what's going to happen, right? We've got more CO2, therefore we've got less medicine, therefore we'll have higher parasite burdens, okay? Well, not so much. It turned out to be exactly the opposite. The parasite load per caterpillar actually went down under elevated CO2. This is not obviously what we were expecting. Yes, the cardenolides go down, but so does the parasite burden. So what does the supervisor do under these circumstances? He yells, go do it again. And so Leslie went back up in 2015, made more chambers, made lots more chambers, put more chambers out, got really big tanks of CO2, grew thousands of plants, scraped her knees getting up and down, transferring insects. I had to buy her coffee. I had to get her help. I even found her a boyfriend. Although she doesn't look particularly happy about it, does she?
It doesn't even look like it.
And what happened? Well, the results were consistent. Yet again, we see this decrease in foliar cardenoid concentration on Asclepias curassavica, consistent with Terry Mattiala's work. A decrease in foliar cardenoid concentration in Asclepias syriaca, again consistent with Rachel Vanette's work. We did see an increase in speciosa. We have no idea why, and at some point we're going to have to dig into that in a little bit more detail. But more importantly, what does this mean for the parasite? The news is still good. Despite these declines in cardenoid concentration, we still get declines in spore load on elevated plants in curassavica, on incarnata, and syriaca, not in speciosa. So the news is still good. It's consistent from 2014 to 2015, so I think that's a cause for celebration. But we still have to explain our contradiction, right? The parasite burden is going down, but the cardenoids, the medicine, is going down. How can that be true? And we started to look at this, and it looks as if the answer has to do with the toxicity of the cardenoids that remain. So not all cardenoids are the same. And it turns out that even though the concentration of cardenoids is going down, their average toxicity is going up. What's remaining and what's increasing in concentration are those lipophilic compounds that pass through cell membranes that are most toxic. And it's those toxic forms we know from previous work that have the greatest effect on parasite load. The way I like to think of it is that here we have lots of medicine. Here we don't have quite so much medicine, but the one on the bottom is much more pharmaceutically active. Right? So we think that's what's going on in our system. So for a change, we actually have some good news. As concentrations of atmospheric CO2 increase, monarchs should not get sicker. This is really, really good news. Even though cardenoids are going down, the most toxic ones are going up, they're becoming more medicinally active, and I think we can relax about that. With a bit more time, I'd like to tell you about the work that Holly Andrews has been doing on water availability, milkweed cardenoids, and the parasite, but I don't have time. I'd also like to tell you about what Joanna and Nefosi's been doing with temperature, but I don't have time for that either. And in fact, all of this is organised below ground by mycorrhizal fungi. We are just nothing but the stumbling child kings on the face of the planet. Our whole lives are organised by mycorrhizal fungi below ground that organise the quality of everything that we eat, including what monarchs eat and the cardenoid concentrations of plants. It's all down to mycorrhizal fungi. I don't have time to tell you any about that. So what I really want to just finish up with is the idea that there are complex interacting factors determining how monarchs are doing. This is still bad news. There's no question it's still bad news. But it's not the CO2. And I can tell you it's not the temperature and it's not the water. And it's helpful to know that because it means we can focus what is important. And on what is important in this system simply is habitat destruction. The best thing we can do to help the monarch is to get on the phone to those people in power and tell them, Monsanto, we can't do this anymore. It's just not appropriate. There are better things we can do than spray our crops with indeterminate broad-spectrum herbicides. We can do better than that. Anyway, just a thanks to the past decade of milkweed research at UMBS here, the undergraduates who participated in our program. Many, many thanks to those individuals, some wonderful graduate students who have helped us understand our system. Great folks, great staff at UMBS. Some call-outs here to people who've helped us particularly, and to Dave Carroll, who's not here, who's provided access to his stuff, and Hillary, who can't make it today, and some major support. That's all I have. I wanted to leave some time for questions, and I'll be happy to answer any that you have. Thank you.
We do have time.
Rex.
I wonder if Monsanto could find in the goodness of their hearts to make Roundup-ready milkweeds.
That's an excellent idea. I think the problem is having someone who's willing to do the genetic architecture of the milkweed. Somebody has tried to develop a sequencing program for the milkweed, so that's actually not out with the realm of possibility. Another way of looking at it, however, is that the monarch, wonderful though it is simply a canary in a coal mine for a bunch of other stuff that's going on. So what we don't see are the pollinators and the spiders and the parasitic wasps all of which matter in these ecosystems, which we're losing as well. But you're right, you could engineer some of the most important native plants as well. It is possible. I think it might be easier just to recognize that we don't need to produce corn and soybean by destroying all the weeds and crops. We don't need to do that.
Stay on corn for a second. So when I was kind of tracking this, the corn price and the conservation reserve program land, the amount of land in conservation reserve, which is kind of bordering farms, were negatively correlated. So I'm wondering if you've ever looked at long-term corn prices related. I know that that's probably really good milkweed habitat and all the perimeters around.
Yeah, there's no question. When Rachel Dennett and I first went up to UMBS, we used to be able to wander around the cornfields in and around the bank station, and within a matter of hours get enough animals for all our experiments for an entire year. We can't do that anymore. Tied to corn prices, almost certainly, and I'm not sure what policy tool would help with that. Frankly, I think listing the Monarch is the way to go, because if we list the Monarch, we basically say you can't spray or have it. But there are probably other policy restraints that we could choose.
What do you think historically was milkweed habitat before it was the margins of fields?
So it's a good question, and the answer is probably early successional patches across the landscape, right? So prior to European settlement, it would have been whatever patches occurred naturally or were created by First Nations. But this symbiosis goes back a long way. So even in the early 1900s, Emmett County, Michigan, had the greatest stem density of milkweed in North America. So it's more than 100 years old that these habitats have been supporting that. Of course, we don't have records from Mexico, the number of hectares covered prior.
Thank you, Mark.